The flower beetle is a bug distributed throughout Southeast Asia. They aggregate on tree sap sites and fruits to feed and mate. Unlike most other flower beetles, females of Dicronocephalus wallichii exhibit nesting behavior before oviposition, which is the laying of eggs. To make a nest, a female constructs a burrow in the soil, cuts dead plant leaves into small pieces to provision the nest, and then lays one egg inside the nest. Hatched larvae have been thought to feed on the nest materials prepared by their mothers, but the effects of pre-ovipositional care on larval performance was not previously tested. In this study, researchers set up an experiment to examine the effects of provision nests on the survival and growth of hatched larvae under both poorly and highly fermented soil environments. They provided 3 conditions. For the nest treatment condition, a nest without an egg was manually buried in the fermented bark, in the same way as constructed by females. For the leaf treatment condition, they provided a mixture of fermented bark and dead leaves. This treatment was included in order to examine whether the processing of leaves by females had a positive effect on larval performance. For the bark-only treatment condition, they provided only fermented bark and no nest or leaves. A newly hatched larva was placed under each of the 3 conditions. 20 days later, the researchers checked if the larvae were dead or alive and measured their body weight. They also reconducted the experiment using poorly fermented bark, thus testing 6 experimental conditions in total. The result was clear. When provided with only poorly fermented bark, 94% of larvae died, and pairwise comparisons showed that their mortality was significantly higher than those under any of the other 5 treatments. No other significant differences in mortality were found. The body mass of larvae provided with dead leaves and poorly fermented bark was significantly smaller than those placed under any of the other 4 remaining treatments. No other significant differences in larval mass were found. In conclusion, the researchers found that feeding on nests enhances larval survival and growth rate in poorly fermented soil conditions. By contrast, in highly fermented soil conditions, larvae showed a high survival rate and fast growth irrespective of the presence or absence of nests and dead leaves. Thus, it seems dead leaves or highly fermented organic matter may be crucial for the survival of hatched larvae. Moreover, they found that larvae provided with nests in poorly fermented soil grew as fast as larvae in highly fermented soil. This indicates that females alleviate the food stress of larvae during the initial developmental stage by constructing provision burrows. If you would like to learn more about this study or see other publications in the Journal of Ethology, please visit the following website.